Hi, my name is Alex, and today I'll be presenting layer-specific deactivation of prefrontal cortex as a causal test of predictive coding. Briefly, in the predictive coding model, prediction signals originate in the prefrontal cortex and are sent to lower cortical areas. Sensory information, of course, originates in sensory regions such as visual cortex and propagates up the cortical hierarchy. Previous work from our lab has shown that prediction signals are carried via alpha and beta rhythms, which feed back to lower cortical areas. These represent our brain's internal model of the world. These alpha and beta rhythms may also suppress gamma rhythms that feed forward sensory inputs. There are also differing roles of superficial versus deep cortical layers. Consider the case of the same cue every trial. This is a very strong prediction signal. The deep layers of prefrontal cortex, or the feedback layers, send a very strong alpha and beta rhythm down to sensory cortices. This strong alpha and beta is then able to suppress superficial layer feed-forward activity, represented by gamma and spiking activity. Conversely, here's a case of a very unpredictable cue, a different cue every trial. There is a weaker prediction signal, so less feedback alpha and beta rhythm to sensory cortices, and therefore less suppression of superficial layer gamma and spiking. Therefore, the sensory input can be fed forward uninhibited. The goal of this project is to test this model by selectively deactivating either superficial or deep cortical layers of prefrontal cortex. To achieve this, rhesus monkeys will perform the delayed match to sample task. There will be 3 different stimuli used as cues, and there will be predicted and unpredicted blocks of 20 trials each. 2 plexon laminar probes will be inserted into each brain area, prefrontal cortex, parietal 7A and V4, and 2,000 nanoliters of 100 millimolar mucimol, which is a GABA A agonist, will be injected at 50 nanoliters per minute over the course of 40 minutes. To achieve layer-specific suppression, I use plexon probes with fluid injection ports at either superficial or deep locations, and I align the probe online to the cortical layers. In this example, we have time in minutes on the x-axis, and we have channel number on the y-axis. This is a 16-channel electrode. The colors represent multi-unit activity as a fraction of baseline. So here, this red horizontal bar represents injection of GABA, which is an inhibitory neurotransmitter, and the blue represents a decrease in multi-unit activity. So this is actually a neuronal suppression. And you can see this neuronal suppression is localized to the area surrounding this fluid injection port. This selective suppression can be achieved in either superficial or deep cortical layers. Now to quickly show some results, deactivation of deep layer prefrontal cortex, and what is the effect on the distant area V4. On the x-axis, we have time in seconds. This first 1 second is when the cue is present on the screen. And on the y-axis, we have normalized gamma power. Control is in black, and the drug mucimol is in blue. So after injection of mucimol, the deep layer prefrontal cortex, in superficial layer V4, we can see increased gamma power. Similarly, we see increased spiking activity in superficial V4. And it's the same story in deep layers of V4, increased gamma power and increased spiking. Next, we can look at deactivation of superficial layer prefrontal cortex. There's a less striking result, no clear change in gamma power. However, there's increased spiking activity in superficial V4. Finally, there's the control experiments, injection of saline or ACSF. You can see there's no clear result in spiking activity. However, there may be increased gamma in superficial layers of V4. It's possible the injection itself is slightly disrupting local neural activity, something I'm investigating further. To summarize, the drug spread can be contained within specific cortical layers. Mucimol to deep layer prefrontal cortex especially seems to increase V4 spiking and gamma. And consistent with our hypothesis, feedback layers of prefrontal cortex do seem to suppress visual cortical activity. Thank you to the Miller Lab, thank you to Simon Center for their support, and thank you for listening.